In this lesson, we are going to look at the overview of what occurs in order for a gene to give rise to a protein. So there's essentially two major parts. It's called transcription and translation. And then there's modifications that occur after. So we're going to just touch on each of those topics. And then our next 3 lessons are going to dive into each of those with more detail. So again, you're going to see a lot of information, and but each lesson that comes after this will have more details. So the original hypothesis was that one gene would lead to one enzyme. And so that was the one gene, one enzyme hypothesis. So it was a hypothesis that each gene is unique, which they are, but the big difference is that they thought that each unique gene would code for the synthesis of a single enzyme. Now they have a better understanding of the genome and the functioning and structure of enzymes. They now realize that there's one gene, one polypeptide. So that's the one gene, one polypeptide hypothesis. So this is where they hypothesize that each gene is unique, so that hasn't changed, and codes for the synthesis of a unique single polypeptide. And we're going to see how a single polypeptide can be modified to create different enzymes. So let's look at the synthesis of arginine to introduce this lesson. So we start with a precursor, and this is any specific molecule, and that precursor gets modified by enzyme 1. This enzyme 1 is actually called arginine E, and it's coded by arginine E gene. And so arginine E enzyme will alter this precursor and create ornithine. Ornithine will be converted to citrulline by enzyme 2, which is coded by ARG-F gene. So we have this gene coding for enzyme 2, which has the function of converting ornithine to citrulline. Similarly, we have citrulline being converted to argininosuccinate. Argininosuccinate. I won't make you say that. And this is done by enzyme 3, which is coded by ARG-G gene. And argininosuccinate is converted to arginine by enzyme 4, which is coded by ARG-H gene. So we have 4 different genes coding for 4 different, in these cases, enzymes. And if we have a mutation in any of these genes, and which would lead to any of these enzymes not functioning, you're not going to get a continuation of this cycle. So for example, if we have a mutation in ARG-E, then we're not going to get this precursor converted to ornithine, and then nothing else would progress after that. Similarly, if we have a mutation in ARG-F, that would be making enzyme 2 not be functional, then synthesis would stop at this point, and so on and so forth. So we have the central dogma of genetics, and this is the law, knowing that we have the conversion of a gene, so shown here. So we have our double helix DNA, and we have the conversion of a gene into what's called an mRNA. So please don't confuse this with mitochondrial DNA or my beloved microDNA. This is mRNA for messenger RNA. And we'll talk about each of these again in more detail later. So mRNA is a messenger because it essentially converted a DNA gene to an RNA fragment of that gene, or RNA version of that gene. That mRNA gets read by ribosomes and translated into an amino acid sequence, a polypeptide. And then this polypeptide, we know, gets folded and modified, we learned about this in Unit 1, to create a functional protein. So this is called the dogma. And dogma is a word to talk about the hierarchy or the order of things. And so this picture is from the movie Dogma, which is one of my favorite movies. And it has Alanis Morissette, who is Canadian, as God. Doesn't get better than that. Okay, so let's start with transcription. So step 1 is transcription. And this is where the information coded in DNA is copied into an RNA. So again, this is copying of DNA into RNA, and it's literally just copying it in the same language. So you're still using nucleic acids. DNA copied into RNA. So DNA is transcribed into an RNA copy. This occurs in the nucleus of eukaryotic cells. Prokaryotes don't have a nuclei. This RNA exits the nucleus through the nuclear pores. The next step is translation. This is where the information coded in the nucleic acids of RNA is copied into amino acids of proteins. So this is where we get the formation of a polypeptide. This occurs on the ribosomes in the cytosol. RNA contains information for polypeptides in the language of bases. These need to be translated to the language of amino acids. So it's translating. You can think of like if we have a visitor from another country and we don't understand their language, say French, because I wouldn't even understand that. I would need to have that translated into English, a completely different language. So we're going from the language of nucleic acids to the language of amino acids. So let's please review the differences between RNA and DNA. So RNA is single-stranded and DNA is double-stranded. RNA has adenine pairing with uracil, whereas DNA adenine pairs with thymine. In both RNA and DNA, we have guanine pairing with cytosine. And then the last difference, RNA is ribonucleic acid, and that name is coming from its ribose sugar, where it has that hydroxyl group, unlike DNA is deoxyribose sugar. So here's a visualization that is from Unit 1, so it's the same diagram that was in your notes. We have our single-stranded RNA, and we have our cytosine, guanine, uracil, and adenine. We also have our ribose sugar here. Our DNA is double-stranded, and we have guanine, cytosine, adenine, and thymine, and we have our deoxyribose sugar, so it's missing that oxygen. Okay, so there are several types of RNA that we're going to look at. So we focus on 3, but I can't help but talk about the 4th because it's my favorite. Anybody who has me as a grade 10 teacher knows. And it's really, really important when we start talking about gene control. So we have our mRNA, which is messenger RNA. We have our rRNA, which stands for ribosomal RNA. We have microRNA, which is my favorite, and we'll talk about its role. And then we have tRNA, which stands for transfer RNA. So we're going to talk about M, R, and tRNAs in this lesson and the subsequent lessons. So mRNA, again, it stands for messenger RNA. It varies in length depending on the gene that has been copied. It acts as an intermediary between DNA and the ribosome. So DNA doesn't leave the nucleus. It gets converted to an RNA version of itself, which leaves the nucleus and meets up with the ribosome. It is translated into protein by the ribosome, and it is the RNA version of the gene coded by the DNA. And so we have this comic where the RNAs are talking to each other. I love that her purse is hanging off of a base. And they are saying, I'm telling you, Carol, I'm done being this guy's messenger boy. He can leave the nucleus and do it himself for all I care. Okay, next we have rRNA. So this is more of a structural RNA. We don't really see it in action in this year. You'll learn more about this in university. But it actually makes up part of the ribosome, and it binds with proteins to form the ribosome subunits, and they vary in length. Okay, microRNA we will focus on a little bit later, but microRNA is really interesting because it binds to target mRNA. Here's our mRNA here, messenger RNA, and our microRNA would bind to this mRNA and block translation. So it will block protein synthesis. So it actually acts as a gene control mechanism. So they can travel extracellularly, which is cool. And we've talked about this with in grade 10, how the gut microbiome can actually send in microRNA and have an effect on how genes are being turned on or turned off by microRNA. And over 60% of human protein coding genes are regulated by microRNA, so that's quite a bit. They're about 22 nucleotides long. And like I said, microRNA from your gut bacteria and actually from the food that you eat can enter your cells and regulate genes. Lastly, we have tRNA, which is our transfer RNA. It functions as the delivery system of amino acids to ribosomes as they're making proteins. And we're going to see this along the way throughout this lesson and the next couple lessons. They're very short. They're about 70 to 90 base pairs long. And so we see this would be the tRNA here I'm outlining. It's got a clover leaf shape to it. And what's cool is it has this amino acid attached to it, and each tRNA is going to have its own amino acid. So there's a separate tRNA for each amino acid. And we'll, again, look at that in another lesson. It's single-stranded, however, what's really cool is it folds upon itself, creating these hairpin loops. And if you don't know what a hairpin is, it's like a bobby pin. I can show you guys one any time of the day. And they create anti-parallel double-stranded areas. So you can think this is a 5' end or 3' end, and this is a 5' end. So it's still actually following the rules if you go through and look at that. And every amino acid has its own tRNA, as I said. And we will look more closely at the structures of these when we get to the translation lesson. Okay, so let's look at the overview of what happens with transcription and translation, and then we are have our animation and then almost done. Okay, so we have our double-stranded DNA, and we see our complementary nucleotides, and we see ACGTs. And we have the unraveling of the DNA. And either template strand could potentially contain a gene. So it's not always the bottom strand or the top strand. So it depends on the gene, which strand it is on. And we have RNA polymerase that doesn't need a primer will start reading the template strand and creating an RNA version of what it is reading. So it'll still lay down complementary RNA nucleotides. That RNA will unbind from the template strand. The template strand will re-anneal, and we are left with what's called an pre-mRNA. So some things have to happen for this pre-mRNA to be functional or to be able to leave the nucleus, and we'll talk about that. So we have RNA polymerase creates the RNA molecule with a base sequence complementary to one strand of the DNA sequence of a given gene. It follows the same rules as replication, so it reads a 3 to 5 prime on the template strand, so it's growing in the 5 to 3 prime direction. The DNA strand being copied into RNA is called the template strand. So just like it was in our DNA replication lesson. So that's the template strand. The precursor mRNA is the original unmodified result of transcription. So this is what's being made, the precursor or the pre-mRNA. Pre-mRNA is modified, and we'll talk about that in a bit, and it will then exit the nucleus into the cytosol, where it will meet up with a ribosome. So let's talk about translation. So translation, we have our mRNA shown here, and it's been modified, so we're skipping a step. So we have our mRNA that has left the nucleus, and it is now in the cytosol, and it meets up with a ribosome. Now recall that our ribosomes are made up of 2 subunits. We have our small subunit shown here in orange, and we have the large subunit shown here. And they're actually not always together. So it starts off with just the small subunit, and then the large subunit will eventually join in. And again, we talk about this in more detail later on. So we have the mRNA associating with a small subunit of a ribosome. And then the ribosome is going to read along the mRNA while the tRNA delivers the needed amino acid. So this here, this entire unit here that I'm outlining is a tRNA. So we have our tRNA, and attached to it is a methionine amino acid. And then next to it we have a tRNA with a phenylalanine amino acid attached to it. So the amino acids end up getting joined together, and we'll see what that looks like in our animation. So not going into too much detail, but again, I want to review. We have a small ribosomal subunit with our mRNA, and we're going to get our first tRNA that binds to the first site, so the AUG. And that site is always how it starts. It's always going to be an AUG. So August babies, we can remember this. There's not many of us, I think. We can remember this because it's always going to be August as a start. So August was the start of a good time in 1981. And our tRNA is going to have this methionine attached to it. So AUG codes for the methionine amino acid. Once this is created with the small subunit, the mRNA, and our methionine tRNA, the large subunit joins in. So that's actually what it adds to it. It's like, okay, you're all ready, now I'll join the party. And the next tRNA comes in, so we have the UUC, which codes for phenylalanine. This comes in. What will happen next is the methionine gets added to the phenylalanine. This tRNA is empty, so it leaves. And then we get the next tRNA. So CGA codes for arginine. You don't have to memorize those, you'll have a chart. And then this growing polypeptide will get added to the arginine. And we're going to see what this looks like in the animation. Now, the last thing I want to point out before we move into the animation is we have, it's shown in this section right here. We see this EPA, and I don't want you to confuse that with what amino acid that's coding for. Those aren't the letters for those amino acids. These are referring to, there's 3 sites on a ribosome. This is the exit site where the empty tRNA is going to exit. P stands for the polypeptide site, and this is where the growing polypeptide is. And then A stands for the amino acyl tRNA, amino acyl tRNA, which is what this is called. Any tRNA which is carrying an amino acid is an amino acyl tRNA. You can also consider it like an acceptor site. Okay. And so this is the site where the new tRNA is coming in. And so what happens is once this leaves, your ribosome is actually going to move, not any of the other things. So the ribosome moves down, and then this would be in the exit site, this is in the P site, and then a new amino acid comes in. So let's look at the animation. So using the same amino acids so we don't get confused, our amino acyl tRNA carrying the methionine, because AUG codes for methionine, and we'll look at this in the translation lesson a bit more. So the methionine binds to the mRNA. We have our small ribosomal subunit, so it's all set up and ready. So the large subunit of the ribosome comes along. It's ready for the ride. Now we have the next tRNA coming in, carrying phenylalanine. And so this is in the peptide site, and then this is going to be joining the amino acyl site, joined here. And again, we have the UUC, which is coding for phenylalanine. An enzyme is going to transfer methionine to the phenylalanine. So we're always going to have the left going on to the right, sort of how we read. And the ribosome moves as those tRNAs stay in place. So that changes this now in the exit site, and now this is in the polypeptide site. And we have a new amino acyl tRNA coming in with arginine, and it's going to bind to the A site. E leaves the empty tRNA, exit site, empty tRNA, EE. And we have the transfer of the growing polypeptide to arginine. Then the ribosome moves along the mRNA, and we will see another tRNA come in and bind at the A site. And then this empty tRNA would leave. So let's talk about these 3-letter codes I keep talking about. This codes for this, AUG codes for methionine. This is new to us. So the DNA we know is a 4-letter alphabet, A, T, G, and C. But there's 20 amino acids. So what's really cool is we have this genetic code, which is the 3-letter code that will code for specific amino acids. So for example, like I said, AUG is the code for methionine. So when a ribosome sees AUG, it's going to be calling in the methionine tRNA. And this is called a codon, the 3 base pairs that code for a single amino acid. And this is read in the 5' to 3' order of the mRNA. So the mRNA is going to be read from the 5 to 3' direction. What's really cool is the genetic code is mostly universal. So there are some other codons that are really special. So all of these definitions you'll need to have an understanding of. So any codon that codes for an amino acid, like AUG codes for methionine, that is called a sense codon. So you can think of, this makes sense. I can follow this chart, it makes sense. And the chart is the chart you were given for this lesson. So a start codon or an initiator codon is the first codon translated in a polypeptide. So in our example, it was methionine, and a lot of the times it is methionine is the first codon. So it's called the start codon. So again, I said August was the start of something good, meaning AUG codes for methionine, which is often the first codon in a polypeptide. Now, stop codons are also called nonsense codons. So they don't specify an amino acid. When a ribosome reaches these codons, the polypeptide synthesis stops. And what's interesting is there's a lot of synonyms in the genetic code, except for methionine and tryptophan. And we'll talk about this when we talk about our translation lesson. So this, I just want to show you briefly how to read these charts if you forget. So you were given a chart, you were given 3 charts. So the wheel is really cool. I want you guys to color that, do a color coordinating, something like this. So pick a color for the letter and then go from there. It just really helps brighten the wheel and helps you follow it. And you can do the same with the charts too. So this is showing you our first codon, or first letter of the codon, second letter of the codon, and then third letter of the codon. And then you can just read it like you would almost something like battleship. So if our first letter is U and our second letter is C, we're in this category. And then you look at what the third letter is. So if you have a UCC, actually if you have UC anything, if you see anything, it's serine. And then I have the full names here for you to refer to. It's not mandatory that you learn them, but if you're continuing with biology, you might as well do it now. It sticks with you, guys. It sticks with you. It's one thing I definitely never have to review when I'm teaching this. And then, so we look here, here's one that's different. So we have anything that starts with an AU, it could be isoleucine or it could be methionine. And methionine is just AUG. Whereas isoleucine, you could have AUU, AUC, or AUA, and you're going to get an isoleucine amino acid put in that polypeptide. So that is the overview of transcription and translation. And our next lesson is going to look specifically at transcription. So where we get the conversion of a DNA gene into a messenger RNA. And then after that, we're going to look at how mRNA is translated into amino acids through ribosomes and creating a polypeptide.